the video lecture for Chapter 3, which is aerobic metabolism or oxidative metabolism. Before we get into the chapter, let's look at your Chapter 3 assignment and go over those questions so you know what to look for when going over these videos. So for our Chapter 3 assignment, first read the directions. They're the same directions that were in the Chapter 2 assignment, which just means that everything has to be typed in complete sentences, no listing or bullet points, and then answers cannot be directly copied from the PowerPoint slides. So first is short answer questions. We're going to describe the structure and the role of the mitochondria during energy production. So that's going to be discussed in part 1. That'll be the first kind of topic we talk about. Next is the electron transport chain, which is part of aerobic metabolism, and how this system generates the greatest amount of ATP. And then describe the steps that allow for that ATP production. Next is factors determining which food substrate will be used to produce ATP during aerobic exercise. So this is, when I say food substrate, I mean carbohydrates, fats, or proteins. So what factors determine which macronutrient will be used for ATP production during aerobic metabolism? Next is describing why is it advantageous for aerobic athletes to have a higher lactate threshold during aerobic exercise. So we'll talk about lactate threshold and we'll describe what factors influence lactate threshold, and you can answer that question based on that. And then last is our critical thinking question, which means you can't just write down kind of definitions. You can't just write down like a description. It's more of an explanation. So what adaptations would you expect to find in the muscle tissue after chronic aerobic training that you would not see with anaerobic training? So this does require you to know the difference between aerobic and anaerobic training. And then you also have to know the adaptations and how those are different for aerobic versus anaerobic training. And then describe what we see with aerobic exercise versus anaerobic. Okay, so after reviewing those assignment questions, we can now get started with these videos. So Chapter 3 is broken up into 3 parts. So there will be 3 videos for aerobic metabolism. So let's get started with part 1. All right, so with oxidative or aerobic metabolism, we know it's the primary energy system used in, you see this picture down here, any long duration aerobic event, such as running a marathon. Before I continue describing this system, let's go over our objectives. So we'll understand ATP producing capacity. We'll look into the role of mitochondria. We'll define the role of oxygen. We'll know different types of training and how this will enhance aerobic capacity. We'll look at the adaptations that allow for more ATP to be produced. We'll look at indirect versus direct calorimetry, and then we'll explain the metabolic role of recovery. So in part 1, what are our main topics? So obviously just talking about the kind of a overview of aerobic metabolism. And then also, in order to understand aerobic metabolism, we have to do a brief review of anaerobic metabolism. We'll look at the function of mitochondria, and then we'll get started with the first system in aerobic metabolism, which is the Krebs cycle. So let's start with kind of looking at the primary metabolic demands of various sports. So this chart and the chart that we'll see on the next slide will be really helpful, and I know this is a long ways away, but your extra credit assignment, it's at the very end of the semester, and it's going to be about you choosing a sport, and you're kind of writing about that sport. And there's one question on there about the different energy systems used in that sport. And so I have this chart here that shows kind of the use of the phosphagen system, which we learned about in Chapter 2, and then glycolysis, also in Chapter 2, and then aerobic metabolism, which we'll learn about here. But this just kind of shows you all the different sports and which ones rely the most on which system. So if you look over here, we see most anaerobic sports like baseball, basketball, boxing. You can go kind of all the way down and you see that most of these are going to be high until you hit marathon, which is low. And then you kind of look over here with aerobic and you see which ones use moderate, like boxing, field hockey, ice hockey, lacrosse, martial arts, and then high for marathon. So I'm not going to kind of go over this chart. It's just a really neat one to look at and see which sports rely mostly on which system for energy production. And then the next slide here shows we see skiing and soccer, swimming, tennis, volleyball, weightlifting. So which ones use high phosphagen versus which ones use lower, so more aerobic. This chart, you'll also may see a question on the quiz or the next quiz or exam, which may have a bunch of sports listed, and you have to choose, which is the primary energy system, so something along that. So this YouTube video right here is, if you're interested in watching it, it just kind of goes over the different energy systems and how they produce ATP for different sports. So it's a really neat YouTube video if you want to watch that. All right, so let's get started with the benefits of aerobic metabolism. So this slide is kind of saying if fuel is metabolized aerobically, and fuel meaning carbs, proteins, and fats, what are the advantages of this? So first, there's no acidic conditions. There's no ATP hydrolysis, which there was in anaerobic metabolism, and there's no kind of lactate accumulation with strictly aerobic metabolism. So there's no acidic conditions, meaning that we can exercise for a longer duration without having to stop due to acidity-induced fatigue. Next is we can use carbs, fats, and proteins. CHO, if you see that, that's an abbreviation for carbs. We see fat, and then PRO, protein. So where glycolysis can only use carbohydrates as a fuel source, this system can metabolize fat and protein. We have a greater amount of ATP production per glucose molecule or per amino acid or free fatty acid. So greater capacity, but less ATP produced per second. So ATP is produced at a slower rate, but we have a larger amount of it. So it's a more efficient system for longer duration exercise. And then the waste products are nothing acidic. They're innocuous, which means they're harmless. So water molecules is one waste product, which is used by the body. And then carbon dioxide, so CO2, which is transported in the body and then expired by the lungs. So these products can be used or expelled. They're innocuous, they're harmless, so they don't cause any acidity. The body can use the water or exhale the carbon dioxide. So this is also the primary energy source used at rest and then during extended low intensity activities. So because of its high energy producing capacity and absence of an acidic condition, which is a performance limiting substance, there's ample amount of oxygen for the body's tissues over time. So we have lots of oxygen. So anytime we're at rest, we're sitting down, you're watching this lecture, your body's using aerobic metabolism for energy because the energy demand is not high. It's very low. All you're doing is sitting down or laying down, resting. So you don't need a lot of ATP per second. You need a lot overall, but not at a fast rate. So that's the primary system that's used. Or it's used in low intensity extended period or for extended periods of time. So such as long distance cycling or slow jogging for, a few miles, where the intensity is low and we can perform that activity for hours, but we need lots of ATP at a slow rate. So yeah, we'll continue to discuss aerobic metabolism, but let's look at this overview slide here. So this is kind of also referred to as energy system number 3. So aerobic systems, it's also called oxidative phosphorylation, which we'll discuss that term later. So we see energy source, combustion, which means destruction or use of carbs as glucose or fatty acids in the presence of oxygen. We see kind of this process here, which we'll learn about, so I won't spend time on it, but we take glucose, put it through glycolysis. We already know that phase. We get pyruvate. Now we're going to talk about how pyruvate goes through the Krebs cycle and then the electron transport chain. High capacity, low rate of ATP. So we produce a large amount of it, but very slowly. We'll learn this right here later. And then contribution, dominant energy system for maximal intensity exercise of a long duration, so plus 3 minutes, or sustained lower intensity exercise like a marathon. But, let's continue talking about Krebs cycle, which is the first part of aerobic metabolism. Krebs cycle is also referred to as the citric acid cycle, but in this class, I do just refer to it as the Krebs cycle. So let's talk about what are the main roles of this. So we produce some ATP, a small amount, but it's useful. And then we oxidize substrates and remove hydrogens. So oxidize means to become chemically combined with oxygen. Oxidation is also the process of losing electrons, and also kind of the process of taking away or removing hydrogens. And so we'll talk about how this system oxidizes our substrates by removing its electrons and removing its hydrogens. Next, we already know that derivatives of carbs, fats, and proteins can enter into this system, and so we'll talk about each of those processes. And then the electron transport chain, you'll see it referred to in this lecture as ETC. So we see NADH and FADH, those are going to transport hydrogens to the ETC. We already know the role of NADH. We learned it in chapter 2, but it's just a hydrogen carrier molecule, and then FAD is also a hydrogen carrier molecule. So they're going to transport those hydrogens kind of throughout the ETC. Next, we see function is to produce the majority of ATP via oxidative phosphorylation. So this is the final stage in cellular respiration where cells use enzymes to oxidize nutrients, releasing energy to form ATP. The electrons in this system are going to be passed from one molecule to another, and energy is released in the electrons as they are transferred to make more ATP. So we're basically harvesting electrons from carbon compounds like carbohydrates to use, and that allows us to make more ATP. Oxygen, so we see here O2, oxygen acts as the final hydrogen and electron acceptor by forming water. So combining oxygen, which is taken in through the lungs, with 2 hydrogens will equal H2O. And so we take half an oxygen molecule, 2 hydrogens, gives us H2O, which is water. And we'll learn all about that after we get to the very end of the ETC. So before we continue with learning about the ETC, we have to understand, where does it take place? So it takes place in the mitochondria, or when we say mitochondrion, that's singular, meaning one mitochondria. So we probably already know that mitochondria is the powerhouse of the cell. This is where the most ATP or energy is produced. They are organelles that are responsible for aerobic metabolism. So I have a picture here. This is of a muscle fiber. So this is a muscle fiber, and then inside the muscle fiber we have these myofibrils. You're probably kind of confused at this point because we haven't learned about skeletal or the muscular system yet, which is in chapter 4. But we can kind of grasp it. We have all these muscle fibers in our muscle tissue, and then all the muscle fibers are made up of myofibrils. And we see myofibrils, and then we see these organelles that kind of surround each of the myofibrils. And what those organelles are, if you see right here, that's the mitochondria. So that's what we're looking at. We're looking at this structure. So So glycolysis, which we know takes glucose and turns it into pyruvate, occurs outside the mitochondria in the systole of the cell, or kind of the sarcoplasm, cytoplasm, which is just like the surrounding part of the muscle fiber. And then we'll talk about something called beta oxidation, which is the oxidation of fatty acids, which produces acetyl-CoA. So we'll discuss that part. And then Krebs cycle, citric acid cycle, same thing, occurs in the mitochondria. And then oxidative phosphorylation, or the electron transport chain, also occurs here. So as you can see, there are many working parts in a singular mitochondrion that will work together to produce ATP throughout metabolism of fat or of carbs or glucose. Let me give you some more facts about mitochondria, which is kind of interesting, and you can write about this in your assignment if you would like to. This can apply to number 1, but it can also apply to the adaptation number 5 of your assignment. So for mitochondrial biogenesis, which is the formation of more mitochondria in a single cell, so in a structure like this, endurance exercise has to be performed regularly with balanced increase in duration and intensity. So performing long duration aerobic exercise over long periods of time. This can result in an increase in efficiency of the mitochondria to meet energy demands of exercise. And also can result in an increase in the number and size of the mitochondria to produce more ATP. When we exercise, all of these mechanisms right here are ramped up or amplified very extensively by up to 200 times more than when we are at rest. So exercise, these systems are working 200 times as hard to supply energy for the body. So pretty interesting. Let's continue with mitochondria and discuss kind of the structure of this, what would we call it? A organelle. So outer membrane. So the outer membrane, special proteins known as porins right here, and we see that little dot, which is our porin. This is an integral membrane protein that allows movement of molecules to pass through it. So it's restrictive on what molecules can pass through or into the mitochondria. The outer membrane, which is just this outer structure here, is made of lipid and protein. We know lipid is fat, and then protein. So these structures are important, the outer membrane, but what's going to be more important is our inner membrane, which is tightly regulated. Basically the inner membrane is comprised of or composed of something called cristae. So cristae is the inner foldings. So we see all these different folds throughout. These inner foldings create a greater surface area and increase functional capacity. They're sensitive to factors such and stressors, including diet and exercise. They're tightly regulated as it requires a host of proteins to regulate what comes in and what comes out of the cristae. They're less permeable than the outer membrane. So the outer membrane is pretty permeable to substances entering into the mitochondria, but the inner membrane is tightly regulated and only allows oxygen and ATP to pass through this inner membrane here. It aids in ATP production, so this is kind of where all of that ATP comes from. The intermembrane space, which we see here, is the space between the outer and the inner membrane. It's the same composition as the cell's cytoplasm. And then the matrix, which we see here, matrix is just kind of inside the cristae, inside the foldings, all these structures here. This contains ribosomes. So ribosomes, protein complexes that serve as a site for biological protein synthesis in the translation of RNA and DNA. So it's our genetic material. This here is a picture of what a real mitochondria looks like under a microscope. We can kind of see all the structures that we just mentioned. This YouTube video here goes into mitochondria in more detail, so you can watch this, click on it on your slides on your own to get more information. So next, we do need to do a quick review of anaerobic metabolism, and that will lead us directly into aerobic metabolism and kind of continuing with energy production. So the ATP-PC system, or phosphagen system, we know lasts for about the first 15 to 30 seconds of exercise. We're using the stored ATP and creatine phosphate for energy during that time. So we have those storages in our muscles. We know it's about 100 to or 100 or so grams of up to maybe 400 for PCR of those sources. But we do know it depletes quickly within the first 15 seconds or so. And then kind of as we move to the next 30 seconds, we have two options here. We need a recovery period before we can do another really high intensity bout. So let's say I did, burpees or squat jumps for 15 to 30 seconds. Well, my energy runs out if I'm doing it at a really high intensity. So I have to take, let's say, like a 30-second or a 60-second break to get back some of this ATP and creatine phosphate. And then I can do another bout of exercise. Or if I want to continue exercising without stopping, I have to lower my energy rate, and then glycolysis becomes the primary energy system, where now I can use carbohydrate sources for energy. So basically, I either have to rest to resynthesize ATP and creatine phosphate for my next high intensity bout, or glycolysis will kick in to help with help of aerobic metabolism, depending on the length of the rest and how many bouts I'm performing. So yeah, we kind of have two options here, and then let's say we do move into glycolysis. So we know glycolysis starts up or now becomes the primary energy system after 30 seconds and lasts up to 2 to 3 minutes. And it's going to use carb sources for energy. We already know the process of glycolysis. We're taking glucose or glycogen and producing pyruvate at the very end. We know that we produce 2 ATP if we use glucose and 3 ATP if we use glycogen. And then that also results in 2 NADHs. Now we have two processes that can occur. So lack of oxygen in the cell, pyruvate is going to be converted to lactate via fast glycolysis. So at high intensities, pyruvate and NADH increase above what can be handled by anaerobic metabolism and convert it into lactate. So this increases our blood lactate level and may cause acidity. Or we move into slow glycolysis. So slow glycolysis, lower intensity exercise, this avoids lactate production where pyruvate can now enter the mitochondria, but it must be converted into another substrate to enter into the Krebs cycle. So it has to be in an acceptable form, and that's what we're about to learn in this picture right here, and we'll also review it one more time on the next slide. This process is called the intermediate step. This is in between glycolysis and the Krebs cycle, where we take pyruvate and we turn it into acetyl-CoA. So we have 2 pyruvate molecules at the end of glycolysis because we had, because the process or the system split, glycolysis split into 2, resulting in 2 pyruvates at the very bottom. So that's where these 2 pyruvates come from. The goal is to turn the 2 pyruvates into the 2 acetyl-CoAs. The enzyme that will do this is called pyruvate dehydrogenase. So this will kind of facilitate this reaction, help the reaction to occur faster. Okay, so let's discuss what's going on here. So 2 pyruvates, those are a 3-carbon molecule. So what happens is when broken down, 2 CO2s are given off here. So we lose 2 carbons and the oxygen that's combined there. So we can just exhale this. So that's a waste product. So we exhale the 2 CO2s that are given off. We also lose a carbon group, or basically the carbon group we lost was combined with oxygen. So with the 3-carbon molecule, we lose a carbon, 1 for each of them. So it says 2, but remember we had 2 pyruvates. So each pyruvate lost a carbon and they combined with oxygen. So we have 2 CO2s that were lost. And then in this process, NADH is going to pick up the released hydrogen. So hydrogens are released during combustion, and so the hydrogen will break off, and it's going to be picked up by the NAD, which will turn into two NADHs again because we had two pyruvates. So two carbons are left over. So we lost a carbon. So now we have two carbons left over, and we see those right there. So two carbons, and then this coenzyme A is added to the two carbon molecule to form acetyl-CoA. So one acetyl-CoA one pyruvate equals one acetyl-CoA. Since we had two pyruvates, that equals two acetyl-CoAs. So two carbons carry over, combine with coenzyme A to form acetyl-CoA. So what is a coenzyme A? We already know what a coenzyme is. It is present in a lot of reactions that just help the enzymes function better. And they can also combine with carbons and other molecules. So coenzyme A plays a role in the synthesis and oxidation of free fatty acid. So that's kind of what a coenzyme A is, and when it combines with carbon, it forms acetyl-CoA. So let's go over this process one more time before we move on. So this picture, some students like it better because it kind of breaks down the carbons and the oxygens and hydrogens and everything. So this is our intermediate step. We have pyruvate to acetyl-CoA. So this only occurs if oxygen is present in the cell. So a carbon group is removed from pyruvate, releasing carbon dioxide. So we see over here, we have a 3-carbon molecule, and then there's our hydrogen. So we see here that with our pyruvate, we lost one of those carbons. One of those carbons split off, and it combined with oxygen because we know oxygen is present. So we just exhale that CO2. Also, we see this hydrogen here. One of the hydrogens is going to be lost during that process. It's going to break apart. And when it breaks apart here, it's going to be picked up by our NADH. We already said pyruvate dehydrogenase is the enzyme that results in, or kind of facilitates this reaction. And now we have 2 remaining carbons that are going to transfer over here. So we see our 2 carbons that are left. And then we add a coenzyme A. So there's our CoA right there. So the electrons are produced or transferred. So the electrons that were produced during this combustion transferred to the NAD, which will turn into NADH. We can pass the NADH to the electron transport chain, which we'll discuss later. But yeah, that's how we get pyruvate to acetyl-CoA. It's basically just a transferring of molecules. We're losing carbons, we're losing electrons, and we're adding the coenzyme A. And so that's how we just kind of turn into this other structure here. The process occurs twice for each pyruvate molecule. Or 1 for each pyruvate. So we basically end up with 2 acetyl-CoAs because we started with 2 pyruvates. So that's the intermediate step. We take pyruvate, turn it into acetyl-CoA. Now acetyl-CoA can enter. That's the intermediate step right there. So everything I just went over, you can see that it occurs before our little Krebs cycle right here. So our intermediate step, again, last time I'm going to talk about it, is 2 pyruvates that came from glycolysis. We see here the coenzyme A was added, and we lost a carbon dioxide. And then the NAD picked up the electrons that were passed off during this process, and that gave us an NADH. And then we see here acetyl-CoA is now going to enter into the Krebs cycle because it's in an acceptable form. So the only kind of byproduct we got from this was the 2 NADHs, which is beneficial because that'll help us produce more ATP later. Okay. So normally we would do this in class at the end of our lecture, and you would draw out the steps of glycolysis and then add the intermediate step. Again, I think drawing out stuff is really important for learning. You don't have to do this, but it is kind of suggested because knowing this process is really important. I had to do this in my undergrad and in my master's when I was taking bioenergetics. So drawing the steps of glycolysis, knowing where everything is, and then adding that intermediate step. Just knowing what happens, the important enzymes, conversions, NADH, and ATP production. But let's move on to the Krebs cycle. So the Krebs cycle is named after Hans Krebs, who identified the steps in this pathway in the 1930s. So we've had a lot of history, almost 100 years of teaching and learning these steps. So let's talk about what's going to happen here. So we have to recall that word oxidize because a lot of oxidation is going to occur. So I said that oxidize means to chemically add oxygen, but it also means to undergo a reaction in which electrons are lost to form another product. And that's what's going to happen when we kind of see all these transformations from acetyl-CoA into other products. They are undergoing reactions, losing electrons to form something else. So first, breakdown of pyruvate forms acetyl-CoA. Now we enter the Krebs cycle. So acetyl-CoA will combine with oxaloacetate. Oxaloacetate begins the cycle and is regenerated after the 8 steps. That's why the Krebs cycle is a closed loop, like you saw on the last 2 slides. It's a circle, basically it's a cycle that starts and ends with oxaloacetate. So oxaloacetate will form citrate. So acetyl-CoA plus oxaloacetate equals citrate. So in the presence of oxygen, acetyl-CoA delivers its carbon, so it's 2-carbon acetyl group, to oxaloacetate to form a 4-carbon molecule. So if you remember, pyruvate was a 3-carbon molecule, and then we lost a carbon. Acetyl-CoA is a 2-carbon molecule, and then when it combines with oxaloacetate, that's a 4-carbon molecule. So when it forms citrate, that's now going to be a 6-carbon molecule. We also lose the acetyl-CoA in that reaction. So that's how we get citrate. And then citrate will then go through a series of reactions resulting in 2 carbon dioxides and 1 ATP. We also have 3 NADHs and 1 FADH produced after the end of 1 cycle. And then we times that by 2 because we had 2 acetyl-CoAs and the cycle occurs twice. So we double the products. So at several points in the cycle that we're about to go over, these products will arise. Hydrogens and its associated electrons combine with the carrier molecules, NADH and FADH, and they're moved to the electron transport chain. So even though they're not an energy molecule, they will produce energy in the next system. All right, so here's our Krebs cycle. I'm going to go through each step, and I'm going to give you a lot of information here. So you do want to make sure that you're either writing this down, listening to this very attentively, so you know exactly what's occurring in this system. Okay, so acetyl-CoA is where we left off. We kind of already talked about how we get citrate here, but we see here comes oxaloacetate. So oxaloacetate will come, acetyl-CoA will combine with it. In that process, we lose the acetyl-CoA, the CoA group, but we have the acetyl. So we have oxaloacetate combines with acetyl, we lose the CoA, and that forms citrate. So citrate right here will kind of continue through this process. So this was the first step. So step 1, acetyl-CoA combines with oxaloacetate equals citrate. Now let's move to step 2. So step 2, citrate will lose 1 water molecule and gains another as it's converted into its isomer, which is isocitrate. We already know what an isomer is. It is where 2 products have the same formula, but a different arrangement of atoms. So the exact same formula, different arrangement of atoms. So that's an isomer. And citrate and isocitrate are basically identical. Isocitrate dehydrogenase is going to be the enzyme that kind of helps with that conversion there between citrate, isocitrate. So that enzyme works right here. And then step 3 is isocitrate to alpha-ketoglutarate. So from isocitrate, it's going to be oxidized, producing a 5-carbon alpha-ketoglutarate. we lose a carbon group here. So from this whatever carbon group this was, we lost one, and the carbon combined with oxygen, and now we have CO2, which is a byproduct that we can just exhale. 2 electrons are also reduced to NADH. So during this conversion, we have a loss of electrons. The electrons are picked up by the NAD molecule, forming NADH, which is moved over to the electron transport chain. So that's kind of what happened here. We have our first CO2 that was lost and our first NADH that was produced. So our next step is going to be step 5. Step 4. So step 4 is alpha ketoglutarate to succinyl-CoA. So alpha ketoglutarate converts to succinyl-CoA, releasing another electron, reducing NAD to NADH, and a loss of a carbon group, forming another CO2. And we have an addition of a coenzyme back to alpha ketoglutarate to form succinyl-CoA here. In this process, we lose more electrons, so turning that into, we just said, the NADH, so we have another NADH produced and another CO2 that was lost. The CoA was added back, making succinyl-CoA now. Okay, now we're on step 5 with this arrow right here. So succinyl-CoA, we now have to get that to succinate. So the CoA, this group was just added to form succinyl-CoA, but now it is lost again. So the CoA group is lost and substituted for a phosphate group. So we know phosphates are high energy bonds. This is a phosphorylation process, which means the addition of a phosphate. So we see here GDP. Let me tell you what GDP is. That's guanosine diphosphate. So guanosine diphosphate is GDP, but we add a phosphate group to make GTP. Now we have guanosine triphosphate. So this is our site of ATP production. So guanosine triphosphate, or GTP, is energetically equivalent to ATP, but it's used more restrictively. So its use is more restricted to MPS, which is muscle protein synthesis. So it's really only used in that process. But here it just shows the GTP, but no, that is the ATP. They're energetically equivalent. They produce the same amount of energy. It's just more restricted. So that's how we get succinyl-CoA to succinate, and we produce 1 ATP right here. Okay, now we have to get succinate to fumarate. So succinate to fumarate, what occurs here? So 2 hydrogen atoms are transferred to the FAD, making FADH. So 2 hydrogens equal the FADH, and we're going to transfer those electrons to the electron transport chain directly. So the energy in this step is insufficient to reduce NADH, but it's sufficient enough to reduce FADH. So NADH is a higher, basically, energy electron or energy transfer, but the energy that was produced here is not enough for NADH to be produced, but it is enough for FADH. So we can still use FADH in the electron transport chain. It just doesn't produce as much ATP as the NADH does, but it's beneficial, so we got that. Okay, so next is step 7, which is this arrow right here. So we have fumarate to malate. Water is added. It's not shown in the picture, but water is added to fumarate, and then malate is produced. So this is a really simple conversion. We just add water. The last step, this is step 8 here, this last arrow. We have to get malate and back to oxaloacetate. So in this last step, the cycle regenerates oxaloacetate by oxidizing malate, resulting in another NADH. So when it oxidizes, we already said it loses its electrons. So it loses its electrons, which are then picked up by the electron carrier, NAD, forming NADH. And now we have oxaloacetate again, which will repeat the cycle one more time. So after one acetyl-CoA enters and goes through these 8 steps, we have 3 NADHs produced in the black circles. We have 1 FADH in the blue circle, 1 ATP, which we know was our guanosine triphosphate. But we have to double the products because we see up here, we started with 2 pyruvates equaling 2 acetyl-CoAs. So now the second acetyl-CoA has to go through the same process. When we double the products, we now have 6 NADH, 2 FADH, and 2 ATP after 2 turns of the cycle. So let's do an overview of ATP production here. So we kind of have to count up everything we produced, all the ATP. So we have to go back to glycolysis. So in glycolysis, we produce 2 NADHs and then 2 to 3 of our ATP, depending on if glucose or glycogen was used. Intermediate step, we didn't produce any ATP here, but we did produce 2 NADH molecules. And then the Krebs cycle, we produce 6 NADH, 2 FADH, and 2 ATP from 2 turns of the cycle. So thus far, we've only been exercising for about 3 minutes. The body has produced a total of 4 to 5 ATP, 10 NADH, 2 FADH. So in order to make more ATP during exercise, we have to turn NADH and FADH into more ATP. So that process is done in the electron transport chain. So now we're moving into the ETC. We basically started aerobic metabolism starting with the Krebs cycle here, but we don't produce enough energy. So we have to send these electron carrier molecules to the ETC to make more ATP so we can continue to exercise for longer than 3 minutes. Okay, so that was part 1 of chapter 3. I know it was a lot of information. Watch it as many times as you need to kind of grasp the Krebs cycle, because I know that's a really difficult kind of concept to understand. But what I want to tell you is the reason that we take acetyl-CoA and turn it in, or combine it with oxaloacetate to turn it into citrate, and it goes through all those 8 steps. It's again to make the cell more unstable. Remember, we're in the mitochondria. So if we're in a single mitochondrion, that means we're in a muscle cell. And we want the cells to give off more energy. And the way to do that is to make the cell more unstable. We make it more unstable by kind of moving through all those different processes. And you noticed it gave off NADH and it gave off FADH because we lost electrons, and it also gave off a GTP. So that's the purpose of going through all of those, and that's called oxidizing. Oxidizing is taking one substrate and producing a product, and in that process we are losing electrons. So yeah, I hope this part made sense. But let's continue with part 2. So that'll be the next video. So yeah, see you in a second.